Good afternoon, everyone. It is my pleasure to welcome all of you to Mazar Auditorium this afternoon and to introduce our speaker, Dr. Marjana Tomic-Canic, who is a longtime NIH grantee. I'm delighted to have this opportunity to welcome her to this campus so that she can experience more of the NIH community than she has extramurally. Dr. Tomic-Canic is a professor of dermatology at the Department of Dermatology and Cutaneous Surgery at the University of Miami's Miller School of Medicine. She's also a graduate faculty member at UM's Human Genetics and Genomics, Molecular and Cellular Pharmacology programs. And in addition is the director of the University of Miami's Wound Healing and Regenerative Medicine program. Dr. Tomic-Canic began her career as a pediatric nurse in a burn unit and credits that early unique perspective to what she brings to the basic science research of wound healing and skin disease those early days. She went on to complete her doctoral and postdoc training at NYU Schools of Medicine before joining the faculty at NYU Departments of Dermatology and Microbiology. From there, she became the faculty at Cornell's University Weill Medical College, where she directed the Tissue Repair Regeneration Program at the Hospital of Special Surgery's Department of Tissue Engineering. She's been an active member and established leader in the wound healing and skin biology community, and she's a member of many professional societies. Also served on a number of NIH study sections and review panels, for which we are grateful. She has also participated in a number of national scientific and clinical panels discussing the need for translational research to improve clinical outcomes of wound healing. Dr. Tomic-Canic's current research focus is the molecular and cellular mechanisms of wound healing, its inhibition, and the long-term goal to bring research results to patients at the bedside. Her research includes human and diabetic models of wound healing, wound genomics, generating primary cells from patients' wound biopsies, and cellular assays of wound healing. She has been continuously funded by the NIH for more than 10 years. In addition to her National Institute of Nursing research grants, she has also received support from the National Institute of Arthritis and Musculoskeletal and Skin Diseases and the National Institute of Digestive and Kidney Diseases. As I believe we will hear today, Dr. Tomic-Canic's lab has identified molecular markers that may predict the clinical outcome of non-healing and also guide surgical debridement of chronic ulcers. This revolutionary technique, currently being validated in an NIH-sponsored clinical trial, may provide the basis for the development of new therapeutic approaches to treat this condition and also reduce mortality, morbidity, and disability associated with chronic wounds. As most of you probably know, the United States spends more than $25 billion, make that billion with a B, dollars annually on wound care. So improved techniques for promoting the healing of wounds would have a great impact on healthcare costs as well. The title of Dr. Tomic-Canic's presentation is Wizardry of Tissue Repair and Regeneration, A Tale of Skin Cells When Their Magic is All But Gone. Please join me in welcoming Dr. Tomic-Canic.
Good afternoon, everyone. Can you hear me well? It is really a great pleasure to be here today, and I want to thank National Institute of Nursing Research for nominating me for this lecture and organizers for giving me the opportunity to share today with you our research and approaches to heal wounds. And if it wasn't for poetry and influence of my daughter, who is a big Harry Potter fan, and my son, who loves graphic design, my title would have been slightly simpler and will actually read Cellular and Molecular Mechanisms of Wound Healing and its Pathogenesis, and that's what I will be talking about today. So the overview of a lecture will be a brief introduction of the skin biology and basics of it, and obviously basics of wound healing, and then I will divide the talk in two parts. The first one will be lessons learned from patients where we took the approach of taking the biopsies from chronic wound patients and really trying to analyze both cellular and molecular aspects of inhibition of healing from them, and lessons we learned about basic molecular and cellular mechanisms. And then the second part of the talk will be sort of flipping the sides and trying to go from bench back to patients and apply what we learn in practical setting. And so the primary function of skin, I've been really fascinated with this organ ever since I started my graduate school is that obviously the goal of skin existence is barrier formation and its maintenance. And that is achieved through really a remarkable perpetual process, if you want, where basal keratinocytes, which are the most proliferatively potential compartment of skin, divide, and then this one cell will turn on and start differentiating, completing this sort of process of terminal differentiation and barrier formation. Through that process, the keratinocytes will lose their nuclei, cross-link their proteins, and die. And that will basically, together with lipids, form the barrier as we see it, and cornified layer. So how is this really achieved? Obviously, we have to thank epidermal stem cells for it. They reside in there are two compartments. One is basal layer of epidermis, where they actually asymmetrically divide and then starts differentiating terminally. And then the second compartment that's equally important for wound healing, they reside in the bulge area of the hair follicle. Normally, in a steady state, these cells really don't participate much in biology of epidermis unless the wound happens. They are responsible for formation of hair follicle and the cycling of hair and biology of the hair follicle. But if the wound happens, the bulge area stem cells will start migrating into epidermis and sort of aid to the wound healing process, and that becomes very important for what I'm going to talk about today. So clearly, skin has this enormous regenerative potential and proliferative potential because it perpetually differentiates throughout lifetime and serves us well. So a small biopsy of human skin can generate enough primary cells in cultures to generate sheets that can cover several baseball fields in surface. And that is currently being used in burn units where we actually grow autologous cells and apply them to patients to actually aid in their wound healing. So if you think about the potential, it's really remarkable of what a skin as an organ does. In addition to that, many of you have heard how this was a landmark year of 2007 when a skin cell scientist independently in two different parts of the world, the Yamanaka and Thompson labs, have come up with a protocol where they can use and use adult skin cell and really turn it into induced pluripotent stem cell or embryonic-like stem cell and now become really a potential to generate different tissues. So from an adult skin cell with 4 different genes, slightly different from 2 different cocktails and 2 different labs, but an adult somatic skin cell can really be changed into embryonic stem cell and then give rise to different tissues and differentiate it into different things. So if you think about this conceptual potential of regenerative medicine and tissue repair that comes from skin, it really is remarkable of how the wound healing process occurs throughout lifetime. And it is really a coordinated cellular process that really aims at nothing else but barrier restoration. What I was fascinating about is that you can take a human skin off the body, put it in the incubator, maintain it on the air-liquid interface, and then if you wound that skin will heal. So this is right autopilot. So the cells really do know how to close the wound and how to really enter this evolutionary conserved process of barrier restoration. You could take this process one step away and then take the skin pieces apart to its individual cellular compartments and grow keratinocytes and fibroblasts in culture, then reconstruct 3-dimensionally that tissue and actually make a skin in the petri dish. And if you wound that skin will heal, as the slides are showing. So you could actually see this leading edge of epithelium crawling over the dermal side and really closing the gap. So the cells really do know what to do on their own without any of our help. And how is this process achieved? The basics are clearly, it's not a simple process. And that is both exciting and difficult when you have to deal with treatment development, because obviously When you have multiple cells and multiple factors, it becomes a lot of more complicated story. So when a wound happens, keratinocytes release pre-stored interleukin-1. And that is important because this is sort of a big signal to the surrounding tissue that the barrier has been broken and the pathogens will be coming in. And I think that if you think about these cells, they're equipped to really very profoundly, robustly alert everybody of their neighbors, if you want, that the barrier has been broken. And in response to that, a lot of things happen. Keratinocytes start moving and proliferating, obviously to close the gap. Fibroblasts start proliferating and lay down the matrix. Endothelial cells start sprouting blood vessels and sort of the form of angiogenesis. Macrophages are moving in to clear up the infection. All of these cells are very, very important, and they're very, very well coordinated in timely fashion in order to close this wound. And of course, how does that happen? Well, through the crosstalk among these cells with wonderful emails and cell phones and all the gadgets that they have, which are called growth factors, cytokines, and chemokines. So they talk to each other, and they stimulate each other in the right way, in a really timed response, in a well-coordinated manner. So how, if we actually deal with this extraordinarily tissue that can heal and perpetually does that through our lifetime, how can we then face this in our clinical side? So we are now in the midst of epidemic of chronic wounds, and so worldwide there are data out there that show that there are every 30 seconds part of the limb, lower limb, is amputated due to diabetes. And I think as this is data from 2005, and as we're coming towards this age now, I think the time is even shorter. So with all this ability that we have, and technology and knowledge and science and everything that we have in our toolboxes, how can we use amputation as a clinical approach? That is simply not acceptable. And so the focus of my lab is really to try to understand why wounds don't heal. In a clinical arena, we are faced with a lot of products that are safe to use, but they do not reach the FDA threshold of clinical efficacy. And very few products have reached that, and they actually don't have all that great successful rate. So clearly we are facing with a tremendous clinical burden that really did not come up with solutions or treatment approaches yet in this big fashion. So we are trying to do anything and best possible we can for our patients, but I think in general there is a lot more that can be done. So the question really is why wounds don't heal, and if you want slightly more daring, why the cells lost their magic, to be in line with the title. And so we decided to use the approach, and I teamed up with extraordinary team of clinical scientists, and one of which is Harold Brehm, currently in Winthrop University Hospital, and one is Robert Kerzner and University of Miami. And you know, these are the incredible physicians who actually treat these patients. And Olivera Stojadinovic, who has been working in my lab for a long time, first as a fellow and now as a junior faculty, who actually participated in majority of the studies that I will show you today. So what we decided to do is collect the biopsies in one study with Dr. Brehm, to collect the biopsies from venous ulcer, non-healing edge of the wound, and really standardize these biopsies by histology and immunocytochemistry and do microarrays. And the study with Dr. Kerzner, what we have done is we selected patients who are non-healers venous ulcers and healing venous ulcers, and then compared the two. So again, we implemented clinical outcome in our design before we actually did microarrays. And so I will show you the summary, but before that I have to make a slight digression, and I think it's towards the students that I really love, and fellows who are yet to face the study section world and grant application world. And this is from my, actually the NR, this is the grant that actually National Institute of Nursing Research have funded, but this was before the one submission before the one that got funded. And basically when I proposed to do global transcriptional analysis of wound healing, one reviewer said it's simply not possible. So this is really a very important sort of dance, and you will receive a lot of reviews in your life from grants and papers. And every single one of them does influence you in many ways. So once you set and check your ego aside and really start looking into the message, well, to me, the message was really clear. He said, you know what, I'll believe it when I see it. And so what I decided is really to do this and resubmit the grant after I have done the initial studies. And of course, the grant got funded, and here I am today because everything I have to say really started with that original grant and the original study. So before the microarrays are beautiful when you show them, and essentially the left three patterns are gene patterns, as you know, and heat maps of non-healing edges of venous ulcers, and this is the control we compare it to, and obviously there is an identifiable pattern of genes that really show and resemble non-healing. And then, of course, when you zoom in, you understand that there is about 1,500 of them that are statistically significant, deregulated in a chronic non-healing wound edge. And of course, I will not go into details, but many of them relate to three important keratinocyte profiles. One is keratinocyte differentiation, one is their migration ability, and proliferation. And I will today talk about proliferation and migration primarily. But when you when you look at that profile, and then you go back and look at the patient samples and histology, what you actually see is that everything we saw in the microarrays is really true. First, you actually find tremendously hyperproliferative epidermis. And when you look and zoom in, you could actually appreciate that there are mitotic divisions suprabasally, which in differentiating epidermis never happens. And then if you look at this cornified layer, which is supposed to be without nuclei, and that cell layer that really needs to protect us, you appreciate that there is a lot of nuclei there. So what this means is that the cells really do not execute properly the differentiation process, but they also don't completely execute this activation process of wound healing where they are proliferating, but they cannot move. So there is an inhibition of migration and loss of differentiation, if you will, at this non-healing edge. And so there is this rift that happens, and almost like a Grand Canyon, there is a big pile of cells that are kind of disoriented in space and time, and they cannot do anything what they are so well programmed to do. So that process really fascinates me, and we really try to explain it why. So as I said, one of the characteristics of keratinocytes is hyperproliferation. And then the first question we wanted to ask is, so what is the fate of progenitor cells in chronic wounds? And we start looking at epidermal stem cells. And I will not go into details of this part of biology, but important to know, obviously, as any stem cells, they are kept in mostly quiescent time. But when a cycling needs to happen, then there are a couple of events and signaling molecules that need to play a role. One is that BMPR1 alpha needs to be downregulated, GATA3 needs to be downregulated, and beta-catenin needs to be induced in order for epidermal stem cell to go from quiescent to cycling. Now, what we wanted to really look into is what happens with all these regulatory molecules in the context of a chronic wound patient. And then what we found out is, if you look at the left side of these two important molecules that are really they're downregulated. They're downregulated in ulcer, and they need to be downregulated if the stem cell is entering the cycling mode and leaving the quiescent mode. So this kind of prompts that maybe there is a shift from quiescent to cycling in the context of the chronic wound environment. And then when you look at the second part of the of this equation, whether there is activation of beta-catenin, we actually found a profound induction of nuclear beta-catenin in a non-healing wound, and induction as a consequence of CMYC, which basically leads to hyperproliferation of the epidermis. And what's interesting about it is that long before we actually start looking at this, Dennis Rupp's lab and Fiona Watt's lab have shown that if you overexpress CMYC in epidermis of mouse skin, these mouse will spontaneously develop chronic wounds. So clearly, this pathway does play a role in the context of it, and so many years later, when we start looking at patients, we kind of get into the same story. And which is always nice to get a confirmation of this. And so we have a hyperproliferative epidermis. Now, as a side part of that, so you have a hyperproliferative epidermis, okay, but then what was striking to us is the fact that there is actually a complete loss of epidermal stem cells in this hyperproliferative epidermis. One of the markers is K15, which is keratin 15, and this is immunostaining of normal skin, and you can appreciate the basal layer staining that is completely gone in a non-healing edge of a chronic wound. So this frequent cycling might actually lead to deprivation of the source of the stem cells in the environment of a non-healing wound, because simply all of them went on and became transient amplifying cells. Yet the wound is not healed. And it's not healed because there is no migration. So these cells are piling, but they're not moving. And this obviously happens because of all these signals that are really not really coming. So they're not only migrating in the local, but they also have a loss of homing and loss of different chemokines and cytokines where they cannot call the progenitors to come to the site of the wound. So in addition to not being able to migrate, they also cannot call the cells, other cells, such as circulating progenitors, bone marrow progenitors, endothelial progenitors, or local epidermal stem cells to actually come and aid in wound healing. And so none of that happens. So when we start looking into signaling that controls migration and this whole process, we actually looked at, for example, EGF receptor, and we found really that, interestingly enough, there is almost not a slight decrease, not slight, but a profound decrease of epidermal growth factor receptor in the non-healing wound. And what's interesting about it is that little bit of, I'm not sure whether you can even see, it's actually cytoplasmic. So if you think about what the EGF receptor normally does, it sits on the membrane and receives the signal. And here in a non-healing wound, you really don't have that receptor on the membrane where it should be. So if you apply EGF receptor externally to the patient, there are no cells that can respond to it. So the clinical trial of testing EGF receptor would not make any sense on these patients because there is simply no way of really transmitting that signal even if it's externally supplied. And I think that is one aspect that's very important to understand, that transition for preclinical testing to a human situation, and clinical testing really calls for looking into patient samples much more carefully to make sure that they will be able to respond to whatever we are offering to them. The same thing, and I will not go into details, the take-home message of this very complicated slide, that in non-healing edge of a chronic ulcer, we actually get the same situation where none of the TGF receptors are on the membrane, and not only that, but their signaling molecules are not in place and they're impaired in their function. So at the end of the day, when you look at this, we did a microarray study where we compared, this is Mickey Bloomberg from NYU who collaborated with us, and Irena was the lead author, Irena Pashtar was the lead author on this paper. What happened here is that the genes that TGF-beta normally regulates in a healthy skin or in healthy wound actually are completely oppositely regulated in a non-healing of the chronic ulcer, simply because there is no way of proper signaling of the TGF-beta in the context of a non-healing edge. So another thing, so we are still searching of why is the signaling impaired and how the growth factors and all of that really influences migration. And when you do microarrays, we all kind of have a set of our choices that we are expecting to see and are very exciting to see that it will confirm. But then what we essentially do it for the surprises, and I think that's really the big part of genomics approach, is where you really don't know what you're going to find. And one candidate that we really did not expect was ADAM12 to find induced in a non-healing edge. And this is a molecule that really is very interesting, and it has a lot of parts and functions in a lot of things, but it's actually a disintegrin and metalloprotease domain 12. That's the full name. And it's a sheddase, so it sheds and cleaves off growth factors. That's one of the functions that it does. It also regulates the integrins, and it also regulates a lot of signaling. So when we start looking, we have been a lot surprised, not only that it's uniquely induced in chronic wounds, but also that it's not regulated in acute wound healing process of human skin. So it becomes only present and induced. It normally doesn't function and doesn't do a lot during the regular wound healing, but in a chronic wound it gets turned on very tremendously, and you can appreciate that here on a protein level. Every single panel of these brown ones is actually a different ulcer in different patients, so you can appreciate this tremendous and robust induction of the ADAM12. I have to say it's kind of a bearer of a bad news in general, and if you look at the bladder cancer, for example, you could find ADAM12 secreted in the urine of a patient whose tumor is gonna go metastases. So I think in a way it kind of alerts to the bad situation outcome, clinical outcome. We still don't know exactly how and why in the context of a chronic wound. And the study was done with Carl Blobel from Cornell and HSS. So what we have done is we have generated a transgenic mouse with ADAM12, where we actually knock out ADAM12, and obviously when you think about it, so if a lot of ADAM12 inhibits healing, if you knock it out, you will expect that the cells will migrate, and that's exactly what we get. And this is more visual way of testing the migration on its own when you actually take the biopsy off a skin and put it in, and you actually measure the outgrowth. So it's kind of an inverse version of wound healing, where you're expecting to close, you're now measuring the cells, how far and how wide they can actually migrate. So, and the staining here is showing that these are actually indeed keratinocytes, because this is keratinocyte-specific antibody. So, in addition to what we already knew about chronic wounds that have increased metalloprotease activity, we actually found an increase of ADAM12, and that may be leading to increased processing and growth factors, changes in interactions with extracellular matrix, integrins, and intracellular signaling, all of which would possibly lead to inhibition of wound healing. So the obvious question is, so what regulates all of this, what I told you? It's what are the regulators and what are the main switchers? So we start looking into microRNAs, and I will not really go into details of what they do, but for the take-home message, the important thing is when you have a message, obviously it gets translated in a protein. When you have a microRNA present, that doesn't happen. So essentially, you can actually have a full transcriptional regulation, and you do have expression of the message, and you do have a message being made, but then when microRNA comes, it kind of never allows that to turn into protein. And so we thought that maybe if we approach this genomics profiles that we have from our patients, and if we use the reverse genomics with the help of Christina Leslie from Sloan Kettering, who is really talented computational biologist. So if you take these genes, these 1,500 regulated genes, and you say, okay, if microRNAs are targeting these genes, which microRNAs would be regulated in that tissue if these are their targets and that's how they're expressed? And so that sort of reverse computational analysis led to a list of microRNAs that might be regulated in the chronic wounds. And so with, again, Irena Pashtar and Professor Steve Davis, who we collaborated on this study in Miami, we start looking in the levels of these different microRNAs, and what we found is that they're all induced at different levels, but they're all induced in ulcers. And this is one of them. This is a microRNA 21. This is a venous ulcer biopsy, and you could appreciate this is in situ hybridization, showing this profound induction of microRNA 21. So I will not go into details. There are a lot of data there, but in order to shift to the patients and talk about translational aspects, I will not spend a lot of time on this subject. But it's important that we have identified and documented and proved that these microRNAs that we found induced in patients with chronic wounds really target signaling molecules, and they do regulate their expression, or their protein levels, if you want. And that is one of the profound and important issues with the chronic wounds, and something that we really clinically need to know. So essentially, if you look at the diabetic foot ulcer, for example, what happens to these cells, unlike in acute wound when these cells start migrating, that doesn't happen. Then the fibroblasts really stop proliferating, and they do not lay properly the matrix. Endothelial cells are not migrating, and there is decrease of angiogenesis. Macrophages don't come in the site of the wound in the numbers and ready to fight infection. And what you have is, in addition to all of this, is really formation not only of colonization, but unfortunately of microbial biofilm. And now this wound turns in chronic and infected, and there is a really bad outcome at the end for your patients. And so how is this achieved? Again, the decrease if growth factors, cytokines, and chemokines is one of the reasons why none of this really happens. And so turning from healing to a chronic wound, I'm trying to wrap up of what I have said so far, is that there is an increase of microRNA molecules in the non-healing wound, and that leads to deregulation of the signaling molecules that, as an outcome on a cellular process, has inhibition of migration, hyperproliferation, and loss of local stem cell cycling, and their loss in the area of the non-healing wound, and then obviously loss of differentiation. All of this really inhibits wound closure and epithelialization. And wound is not healed unless it's closed, and that means until keratinocytes do their job, the wound is not closed, and therefore there is no healing. And keep in mind, obviously, that I haven't talked about other components. So we are just in epidermis, so this is only one aspect of the whole process, and I could not list all of the participants, but we didn't discuss today granulation tissue formation in the context of chronic wounds, nor angiogenesis, or this crosstalk of the microbial biofilm and chronic wound. So clearly we have our basic science world completely cut out and work cut out for us. Now, the biggest question there are many labs tackling this and trying to understand it, is why, and what is really what is upstream of all of this? So what regulates these microRNAs? And not only microRNAs, they're not magic molecules, but they do participate. So I don't want you to go home and think, oh, it's easy, we can just reverse these and we are done, but that's not going to happen, right? So I think that understanding what comes upstream of all of this that I have told you will be really important, and that's where the excitement right now is, and there are a lot of work being done on that. But essentially and conceptually, from this healing to a non-healing, right? So you can really think about it in two ways. So either the cell has transitioned and crossed over to the world and it cannot go back. So this is sort of terminal. It's non-healing and there is no way back. Or this cell has underwent really a bad set of decisions and signals and God knows what else, and end up non-healing. But there is a hope, and switches can be reversed and they can really be transitioned over. And actually, both of these are clinically valid approaches. And so when I talk about this first part at clinical meetings, they all shrug shoulders and say, so what can we do today for our patients? And so the current approaches are two, obviously. One is remove the non-healing cells, and the other approach is reverse their fortune. It's very simple if you think about it. So either you can just cut them out and keep those that are actually do have healing phenotype, or you can try to really convert them. This might be maybe more difficult, but then maybe more permanent solution. And so both of them are clinically valid, and they're being implemented today in the clinics. But it's not that easy, really, when you have removed non-healing cells. So what does that really mean? How do I, when you look at the wound, how do you know which one is really non-healing cell and the tissue part, and how you're going to cut it out if you don't know? So we clearly need something more of sophisticated approach to really create this, what we call molecular scalpel, where we can really know and guide the extent of surgical debridement in a chronic wound and really removing this tissue. So if you look at the concept of Mohs surgery in skin tumor cells, so what happens is that when a tumor is in the skin, the Mohs surgeon comes and they cut out the tumor, and then they analyze it on the microscope while the patient is still on the table. They look to see on histology whether they're efficient in removing the bad tissue. So is it all tumor tissue gone? And if it's not, they go back and then resect, and until it's completely clear and histology shows the normal skin tissue, this patient is still in the room, and it's being treated and debrided if you want. So if we borrow that approach and we use that strategy to really try to debride chronic wounds, then we clearly need more, obviously more than just the microscope and the histology, but then we can really analyze this tissue and try to come up with something that will tell us how far to go. And therefore is development of these biomarkers any possible way imaginable. And I will show you the approaches where we are at this stage. But the bottom line is, so if you can tell what is a non-healing tissue, even though it might not happen because we still don't have a really quick method to do this, but if you actually know that you haven't debrided the patient far enough, then you might go and re-debride that more efficiently, maybe as quickly as you can, to really get that patient going. And so is there any biological basis to what I am telling you? The other important question in a clinical arena of wound healing is can we tell when a patient comes in on that first visit whether if we apply standards of care, this patient will go on to heal, or do we have, it will not? And so the question of predictive biomarkers, whether we can really predict clinical outcomes early on, will clearly define how we are going to treat the patient. Because, and I have to spend a little more time explaining you why is this important. Today, what you do is you apply standard of treatment protocol for 4 weeks, and then you're measuring the size of the wound in every visit and dressing change, and then you're looking whether that wound has healing trajectory, and if it heals greater than 50%, that patient will heal with the standards of care. But if by week 4 this does not happen, then biological applications, then different treatment protocol gets implemented, and more biological treatments are being offered to that patient in order for them to heal. So, and this is basically based on a work from David Margolis, who really has shown that this that 4-week outcome is really a surrogate endpoint, and really, really important to really know that. So if we don't have to wait for 4 weeks until we actually help the patients who is not healing, it will be tremendous clinically if we can tell that from the first visit and really start a very aggressive treatment plan from the get-go. And of course, not spend a very expensive biological treatments on patients who really don't need it. So there are two different benefits of this approach. And so what we have set up to do is we decided to collect specimens before and after surgical debridement. and really run all the possible analysis. So in addition to microarrays, we have grown primary cells out of these wounds and studied them in culture, and we also looked at a different variety of analyses on histology and immunocytochemistry. And one thing becomes immediately apparent, and that is that on this non-healing edge of wound healing, of non-healing wound, I told you what happens in epidermis, but this is sort of a dermal side, and you could see that there is a profound level of fibrosis that kind of resolves partially in this post-debridement biopsy, and this is how normal skin looks like. But one thing was striking because, you know, granulation tissue really is always a big problem in the chronic wounds, and I always thought that there is not enough collagen. But when you look at procollagen staining, so the cell basically makes the procollagen and kind of spits it out and lays it out as a matrix. So if you look at the procollagen that's still being made in cell, you could appreciate that there is a lot more cells making collagen in a non-healing wound than a post-debridement biopsy. But yet when you look at the collagen orientation and when you look at the polarized light and how this collagen matrix is laid out, then you can appreciate that really this in a non-healing edge, this really does not, it's not done properly, and this obviously contributes to the lack of migration on the top of epithelium. And again, post-debridement biopsy looks a lot better, and this is how the normal skin looks like. So obviously, striking differences as you would expect in gene profiles. So again, this is a non-healing profile, and it's tremendously different in post-debridement biopsy. And so if you think about, these are all the cells as a part of that wound. And so there is clearly a set of cells that have not only different profile, but different healing potential within that wound. And being able to really reach out to them and find them and stimulate them to really come and do what they're so well known to do would be really tremendous. And if you think about this, it's another aspect of it. We can actually use the gene profile to tell us whether we are far enough, and I will show you a little bit later of how we can achieve that. Now, when you look at the primary cells grown from the wound, that a non-healing edge, you can again. I'm not sure how far whether you can appreciate this resolution, but these cells are really look sick. They don't look normal. They're really big and large, and they don't move at all. And when you look at the cells from post-debridement biopsy, clearly they look a lot like a normal cells. So when you try to test their migration, so I have to say that I was a big believer when you grow primary cells out of non-healing wound and put these cells in all the goods of growth factors and cytokines of the growth media, that these cells will revert right on and there will be no difference when you grow them long in culture. But interestingly enough, not only they don't grow well, but they also do not migrate and don't respond to growth factor signals at all from the non-healing edge. But the post-debridement biopsy is a completely different story. These cells are actually able to migrate, and as I said earlier, and you could, those of you maybe sitting in the first rows, you could appreciate that there is a lot more cells on this side than here, so clearly their proliferative capacity is different and better. So again, this is all sort of contributing to the biological basis of debridement approach, and yes, there is within the non-healing wound, there is a subset of cells that are able to respond to growth factors. They're able to migrate and probably complete the healing. So again, finding that region and finding that debridement margin and distinguish the tissue A from tissue B, non-healing from a healing potential phenotype is really a big question, and that's what we were set to study. And of course, our wizards look slightly different, and this is a wound healing clinic. This is Rob Kersner and Steve Davies and so this is the research and clinical team put together that really is trying to figure out these biomarkers. And every once in a while, you will actually get a slide that looks like this, and that's really quite exciting. And I'm sorry there is not enough light here maybe for you to see, but the non-healing edge, this is a staining for beta-catenin, and what we are looking for is nuclear presence. Nuclear presence is bad. absence of beta-catenin in nucleus is good healing outcome. And so, what we do is we stain the cells and we quantify this in the tissue and really link this to the clinical outcome. And we kind of look at the healing at week 4 or the post-debridement biopsy. But every once in a while you will get a slide like this, and this is the most exciting. You could almost draw the line of the tissue where you actually have a loss of beta-catenin in the nuclei, which will be healing potential, and the other side where actually has all the nuclei present for beta-catenin. So again, there is the debridement margin, and I think it's the matter of how sophisticated we are in finding it and developing the right tools to really get there. But this really gives a great hope. And of course, to transition to the genomics approach, which clearly at this day and age might not be clinically feasible and practical for pure expense aspects, but in general, it's clear that it can serve as an excellent guide to really determine what is a non-healing from healing tissue. And so if you look at, this is the patient, these are 3 different patients before and after the debridement. And again, you could see the profiles, and then this patient obviously had something different. And it turns out that this patient was not debrided far enough, and this is really a non-healing profile. And indeed, when we did all the stainings, and of course when we went back to check the clinical outcome, it turns out that that was a non-healing patient, and the surgeon actually went back and debrided this patient and sort of redid more extensive debridement for this patient in order for them for him to get to heal. But clearly, imagine this is kind of, to some oversimplified version, the pregnancy test for wounds in a way. So you could really use the chips and say, okay, so this is a healer and this is a non-healer. And of course, we don't need a big microarray chip to do that, but what we are doing currently and trying to do is really narrow down the list so that we can have a small set of genes that can always tell when the wound is healing and where is the tissue that really does not. And again, this is just to show you the markers that I have mentioned previous in the talk. This is the EGF receptor. As I told you, it's not on the membrane and it's downregulated. This is the post-debridement biopsy. You could appreciate that there is a restoration of the membranous staining, and the same thing for the stem cell marker K15, where it actually gets restored. Notice, these slides look a lot different than normal skin. So we are not talking about healthy tissue, but we are talking about tissue that has a healing potential. And so to summarize this part is that the non-healing wound, so what we are currently doing is collecting a lot of specimens from patients, and we're trying to look at the healing outcome, to look at the predictive markers. And in a non-healing wound, a lot of them either do not change over time or actually change for worse, such as case in Adam 12 here, or BMPR1 that really did not change. And similarly to beta-catenin, when you look at week 0 and week 4, or K15 or CMYC, and you could actually see here the clinical outcome and trajectory of healing, it's not in place. So this patient clearly is a non-healer. Now, how can we really say that up front is the really one thing that we are trying to figure out. And then again, this is a profile of a healing ulcer, as I showed before, and again, there is a complete decrease of Adam 12 and decrease of BMPR1, and there is a decrease of beta-catenin and restoration of K15 and loss of CMYC, and look at the trajectory of the wound. It's healing. Right? So again, doing this approach with the clinical outcome hand in hand really gives us tremendous potential to search for really appropriate biomarker. But so far, we are narrowing down the list, and we are in a clinical study where we are testing this prospectively and trying really to narrow down both the list of the genes and the cellular biomarkers that can really guide us through this process, clinical process. But I really hope that we will have a gene chip for wounds relatively soon, and hopefully sooner than later. But clearly, our goals are, as I said, which wound would heal up front, and where is the debridement margin, and again, we are now using all the possible approaches. And I want to point out one other thing, and this is again the wonderful world of gadgets and all developments in computational medicine, if you want, that is happening right now. And I want to point out my collaboration, a long-standing collaboration with Dr. Brem, who has developed a wound electronic medical record. So he has a research tool that has that follows 132 clinical variables for each patient. So imagine how wonderful that could be, right? So he's following from the wound size that is measured all the time to every single aspect that is important, any clinical parameter, presence of infection, all the treatments, everything that might be important for the wound healing. So what we are now trying to do is build a platform that can now sort of link this clinical data. This is data that can be exported in the Excel, and it's a searchable Excel table. So imagine if we could now feed all these biomarkers and all these things that we are doing for the tissue for this patient and really connect it with these clinical outcomes and variables that he's doing, and really being able to pin out a subset of patients and really do a more personalized approach rather than this general sort of groups of different non-healing wounds. And of course, one of the apps for that would be wonderful, and that's sort of our goal would be that one day every clinician who treats wounds will have its app, and that app will send a little message and say, okay, there are biomarkers, there is the non-healing outcome coming, please do what your protocol tells you to do. And I think this is the reality, and I think we are there, and that's a really great place to be. Now I will, just a few more minutes, talk about a couple of approaches of the other side. So, and that is really the reverse, how to reverse the fortune of these cells. And obviously, we can circumvent the missing signals by different ways. One is using different approaches, and I have to say gene delivery methods today are really quite well developed, and skin is ideal subject for that simply because it's topical, and especially wounds, because for wounds you don't have to have a long term. You actually have to have gene delivery that will stimulate the healing but doesn't have to stay there 3 years. It can really be temporal. And I think that is really a wonderful place to start really applying gene therapy. Obviously, we could apply a lot of different cells to wounds, and that is different therapies that are both FDA approved and new ones that are being developed. So these are, they could be obviously autologous or not, and obviously different matrix, all of which will stimulate the local cells to really get on the program that they know so well and execute the healing. And so this is one of the projects that we have done, and this is sort of delivery of the vascular endothelial growth factor. These are experiments done in diabetic, different types of diabetes, NOD, which is type 1, and DB mice, which is type 2. And what we have shown that if you treat a mouse, obviously with adenovirus delivering VEGF, it stimulates healing, and it's kind of, yeah, so what? VEGF stimulates angiogenesis, and it was expected that this would be the outcome. But what we were surprised to see is that we have seen a tremendous effect on epidermis. And not only that the wound has healed much faster, but look at how much epidermal layers you actually have here. So clearly there is not only stimulates not only angiogenesis, but epithelialization. The other thing is also the thickness of the granulation tissue, which is tremendous compared to the saline treatment or just a vector alone. And this is again the collagen staining to show how well the collagen is laid out. So clearly a growth factor, yes, but what it does, it really targets multiple cell types. And I think that is one of the approaches that it's important. If we are using an approach that's going to hit a single point, it's most likely not going to be very efficient in a complex scheme of things such as chronic wound. But if there are ways that we can stimulate more than one cell type that all contribute to the wound healing, I think that has a very big value in the chronic wound field. And then the last data slide, if you want, that I will show you is that what we call Dr. Badiavas trial. Evangelos Badiavas is a clinician scientist at University of Miami who has NIH study where he studies a clinical trial of bone marrow progenitors for patients with chronic wounds. And we kind of piggyback on that study, and we come and collect the tissue and grow the cells, and he does the great stuff, which is treating the patients. And one thing that he never thought he would get is Emmy, and it turns out that the University of Miami did a documentary on that's called Stem Cell Therapy Healing Force of the Future, and they won an Emmy on it. It was about this clinical trial and the other one that they do for heart patients in the Stem Cell Institute. But it's you never know when you become a scientist, maybe there is an Oscar in making. But what I want to say is when we collect the biopsies from his patients before and after after treatments with bone marrow progenitors, we grow these cells and then we start following them, looking at different things, and I'm showing here microRNA profiles of them. And you could appreciate that after the first treatment, the response is already good in terms of looking at the expression of microRNA 21 and 106, but 16 is still there. And then you kind of get everything in order after a second treatment. But what this tells me is that there might be a way of what I call ordered restoration. So clearly, you don't have a magic bullet where everything all of a sudden goes back to normal, but there is a sequence of events that can actually lead to a healing process, which in the way of what I told you about the switches, maybe tells us that we can actually revert these non-healing cells slowly but surely through the process back to become a healing. And I didn't even touch upon the induced progenitors because that technology clearly gives a tremendous opportunity also in the chronic wounds as in many other clinical disciplines. But that's, again, another potential of how we can revert the non-healing cells into healing cells. So in summary, I will not read it, but in general, what I have told you is that genomics approach identified non-healing wound phenotype, and what we learned about it, that there is inhibition of cellular migration, that there is a loss of appropriate signals that control growth factors, and obviously lack of differentiation in the epidermis of non-healing wounds. And we have also found that there is a distinct population within a chronic wound that has a healing potential, and that is linked to clear clinical outcomes in patients, and this loss of the signals of control and are really not only coming for what we knew already about high protease activity, but also decrease of receptors that mediate signaling. There are signaling molecules that mediate the signals in the cell, and obviously increase in microRNA molecules. And obviously one approach would be to really debride these cells and restore healing phenotype, or the other one is to reverse their fortune. And I think that this is, I have to say, the probably most exciting times in biomedicine, because I think technologies that we have in our hands are so tremendous that we are yet to learn how to really utilize to advantage of our patients. And I think the good days are coming in many ways, and I'm really excited to be a scientist at this time. With that, I have to say there are a lot of people I have to thank, and it's impossible to list them all. I've been privileged to really work with extraordinary set of people in every institution I've been, and many others, because today collaborations are the key. And I will have to thank, obviously, people in my lab. This is fairly accurate a picture, and yes, this is Miami. I'm sorry. Yes, we do have fun occasionally. And of course, the University of Miami Skin Research Center, which we started recently, and it's really extraordinary set of colleagues. And obviously NIH, because everything I said today really was funded exclusively by NIH, although I do have other projects that are not, but everything I said today really comes from that initial grant from National Institute of Nursing Research that really sort of retained me in the wound healing. It wasn't my first R01, but it was my first in a wound healing. And really I have to thank them for really sort of shedding the light on that path and keeping me in that field. And of course, and most important, thanks to all our patients, because they're a really special kind, and I'm really hoping we might not heal the wounds of the entire humanity, but chronic wounds we hope we will be able to very soon. Thank you very much, and I'll take questions.
Thank you so much, Marjana. We do have time for a few questions. If you will take the microphones on the side and say your name, we can just step up right to the mic.
Well, congratulations for excellent coverage and excellent work.
Thank you.
So you have identified quite a few interesting targets. So how many of them you think you might be able to use it in the patient care? And also a question is, what is the diabetic part of it that accelerates this process of defective wound healing?
Right, so there are two different questions in that question, right? One is why for these, of those 1,500 possible targets. So what we have focused is mainly trying to really identify the potential biomarkers that will predict the outcomes of healing. But apart from that, there are lots of different potential targets that would be validated as pharmaceutical targets. But the issue with that is really, yes, to answer the question very, very shortly, there are a set of targets that can be used. For example, these microRNAs. I mean, they're major regulators, and they can be targeted because they really regulate a lot of what is happening. So they would be the obvious target. But I think that from the microarrays, really, it doesn't point out a single target or a set of targets. It really tells you more about what is going on molecular and cellular level. And really understanding how the cells or tissue got there would be really important, and that's where they're tremendously valuable. And I think that's where the targets are. Because when you take a biopsy of a non-healing wound, that is, if you want, an endpoint, right? That tissue is not healing. But what we still don't know is how the process really sort of, if you turn back the time of the origin when those cells actually progress to the non-healing, I think that would be really important to understand. And that's something that we are working on, is really trying to find out what happened before they actually got to be terminally non-healing, so to speak. Does that answer the first part of the question? It's really, you know, if you ask me, is there a magic molecule? There isn't. There really isn't. And a chronic wound, I don't think that there will be a magic molecule. A single one, definitely not, because it's a complexity of the disease is such that it's simply not possible. I sound like my reviewer, but it's very similar. And somebody here is going to go and prove me wrong, and I would love nothing more than that. But what I wanted to say is that you ask about another thing about the diabetes. Again, in diabetic foot ulcer, there are multiple components. Obviously, metabolic is a very important one, but the other one that goes with diabetes in developing of chronic wounds, there are a couple of things. One is peripheral neuropathy. So patients lose a sensation in their feet, and that really contributes in part. Nobody really knows. I mean, there are groups and labs that are working on this, but we still don't know and pinpoint how much this neuropathy really plays a role and how much is diabetes. They're obviously linked together. But I could tell you one thing. There are sets of patients that develop peripheral neuropathy. They don't have diabetes, but they develop similar chronic ulcers on their feet. So again, there are multiple components, and diabetes predisposes because of obviously metabolic aspects, because of loss of ability to fight infection, and there are a lot of other aspects of diabetes that kind of play a role of why the tissue is not healing. But there has to be something linked to the local, to the feet and the lower extremities, because if you really look at the wounds and surgical wounds on diabetic patients, they actually heal, maybe slower, but they actually do. So it's not, I should say, it's not complete disability of healing, and this, you know, but obviously diabetes is a big part of it. And we have done some experiments to show that young diabetic mice really don't have that much of impairment in healing compared to the aged diabetic mice. So if you think about, it's always some underlying component that also plays a role in addition to diabetes that really plays a role in development of diabetic foot.
Good luck in the challenging arena.
Thank you. One more question, yes.
Hi, I'm Francesca Mascia. Thank you for your nice presentation. I was just curious, does high glucose change the differentiation and motility of keratinocytes?
Yeah, we have done this in culture, and I have to say we haven't seen any effect in culture, but that obviously doesn't mean much. But I can tell you the experiments that we have done in the mice, and that's published in experimental gerontology. And if you look at the mice, the diabetic mouse, young diabetic mouse with controlled or uncontrolled diabetes. So if you look at normal, hyper, and hypoglycemic, you really don't see a tremendous difference in wound closure capacity on these mice. They're all kind of the same. So it doesn't mean that they're good, but they're equally bad, I should say. So that doesn't obviously, again, please, we are talking about mice, and extrapolating this to patients, I really would have tremendous difficulty doing. But I think it's important to understand, but then in aging, it becomes a problem, right? So again, it's a component that really of aging in combination with diabetes in the context of mice wounds that we actually see the difference, but not in the glucose levels. Well, thank you.
Thank you very much to Dr. Tomic-Canic. Also, I would like to invite you to, we have a reception following this talk in the NIH library. So as you exit the auditorium, turn to your left, and you'll see it's right there. So we can continue the conversation there. And the reception is sponsored by us. We thank them very much.